Hello, everyone, and welcome to today's webinar titled How to Quantify 200 Metabolites with One LC-MS Method. I'm Susie Valdez of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is sponsored by Agilent. Now let's get started. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you'd like during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have any trouble seeing or hearing this presentation, click on the Ask a Question box located on the far left of your screen. As a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits tab located at the top right corner of the presentation window and follow the process to obtain your credits. Without further ado, I now present today's speaker, Dr. Kristaps Klavins, Deputy Head of the Proteomics and Metabolomics Facility Research Center for Molecular Medicine of the Austrian Academy of Sciences. For a complete biography on Dr. Klavins, please visit the Biography tab at the top of your screen. Dr. Klavins, welcome. You may now begin your presentation, sir.
Hello, everyone. Thank you so much for the kind introduction, the opportunity to present the work that we've been doing for the biggest part of the last year. So basically what we wanted to do is to really find the method that we can quantify as much metabolites as possible. And I hope that you can find our approach useful and also implement some of the things that we've done here also when you want to set up similar method in your lab or your institute. So to put things a bit more in perspective, what we're doing here, I would like to give also a bit of introduction about our institute. So SAM stands for the Research Center for Molecular Medicine of the Austrian Academy of Sciences. And we are located in Austria, in Vienna, and we are quite privileged to have this really unique location. So we are in the center of campus of Medical University of Vienna, and also there's also next doors a Vienna General Hospital. So we're really embedded in the biomedical research here. And there are 10 research groups here at SAM, and there's around 200 people working here, and we're truly international. So we have people from 48 nations here. And besides research groups, we also have 3 facilities: a sequencing facility, compound screening facility, and proteomics and metabolomics facility. And at SAM, we really pursue the free-minded basic life science research to really achieve the maximum scientific innovation to improve the healthcare. We also train the new generation of biomedical scientists by having a PhD and postdoc program here. And at SAM, what we really stand for and strive for is excellence, quality, and responsibility in everything that we do. And what I really would like to point out is also the super cooperation mode that we're working. So we really try to collaborate everyone with everyone, and as a facility, it also means that we are involved in a lot of the research that's done here in our institute. And Proteomics and Metabolomics facility is a team of 10 highly skilled and dedicated scientists, and what we do, we use liquid chromatography coupled to mass spectrometry to provide analysis of proteins and metabolites in a given biological system. So as I said, our lab mainly uses mass spectrometry, and we have a wide range of mass spectrometers available for metabolite analysis. This includes high-resolution mass spectrometry based on orbitrap technology, such as Q-exactive and Fusion Lumos, and we also have triple quadruple systems from Waters and also from Agilent that I'm going to talk about later. And so this access to this wide range of mass spec instruments really also provides us opportunity to set up different workflows for metabolite analysis. We have workflows where we really try to profile several hundreds of metabolites or lipids. We also have capabilities to run commercial kits for metabolite analysis, and this includes a P180 kit from company Biography. And on the other hand, we also can focus really on particular pathways. For instance, we have assays for TCA and urea cycle, and we can use our high-resolution mass spectrometry capabilities to also facilitate isotope tracing analysis, which we have successfully implemented and used in the past years. And also, we provide our expertise to provide custom analysis for small compounds. This can be metabolites or also small molecules such as drugs. And we are not the biggest facility, so what we have is around 3,000 samples per year. And the major sample type that we see here is the cell samples, as this is one of the systems that we use the most in our institute. We also have experience with biofluids, such as plasma, serum, and urine. We also analyze tissue samples and also organic compounds. So this could be either compounds from organic synthesis or commercially bought drugs. So as you've seen, we have mass specs already, we have experience with them, and the question is, why would we need to another method? So the answer to this question is quite simple. The general trend that we see is that the more is better. The more metabolites you can measure, the more information and value it gives to the biological research. And there is one particular project I would like to point out where it's really essential to have this wide range metabolite analysis provided with a one robust system and instrument. And this project is RESOLUTE. And it stands for Research Empowerment on Solid Carriers. And this project is funded by the grant from Innovative Medicine Initiative, and it focuses on particular protein class called solid carriers, or SLCs in short. And SLCs are the membrane proteins that have an important physiological function. There are around 400 proteins in this protein family, and they're involved in nutrient uptake, ion transport, waste transport, and also in drug transport in the cells. However, this protein family, despite it being so important, is really heavily understudied. There's around 40% of them there are around 40% without known cargo or function. And RESOLUTE aims to really characterize and understand these proteins more in detail. And one of the tools that are used to really characterize and understand them is overexpression and knockout cell lines of those proteins. So you could imagine if you overexpress this protein on a membrane, then more things are transported into the cell. For instance, if it really transports metabolites, you would see more metabolites in the cell, and vice versa with the knockouts. So once you knock out this protein, you would expect less metabolite or transported molecule into the cell. And in this regard, the Metabolomics is really essential tool that can identify those metabolites that are transported with the given SLCs. And also, if we talk about overexpression knockouts, but also one for the vision is that once you overexpress those proteins and you have increased levels of the metabolite in the cell, then also a downstream metabolites of this metabolite, so metabolism products of these metabolites will be increased. So you really want to characterize the whole chain of change that are introduced by overexpressing this SLC. And this is really a large-scale study that aims to analyze several thousands of cell culture samples over a span of several years. So we're talking really of 5,000 to 6,000 samples in a course of 1 to 2 years. And our facility, we were trusted with the task to provide intercellular metabolite analysis for the samples that will be generated during this project. So this really set up some really specific requirements for the method that we want to use for this project. So first requirement was that we need targeted method. So we really want to know which metabolites we measure, and if there are increase or decrease in transport, we know which exactly metabolites are those. We also want to have a quantitative method. So in our experience, quantification is really a good tool to get the robust results and really trustworthy results. Also, requirement was a robustness. So we needed a method that we perform really stable over years. So if you measure the samples now and we measure them within 1 year, we want to be sure that we get exactly the same method performance and exactly the same result, because once again, this is really a large-scale study. So also the coverage of metabolites should be as broad as possible. So not only to identify those metabolites that are transported with SLCs, but also to identify the downstream process or to see how the whole metabolic pathways are perturbed by transporting or by increased or decreased concentration of those metabolites. And of course, this method should be suitable for cells. So we should be able to run cell extracts with this method. Also, what's important, the method we would use for this project should be scalable. in the sense that we can measure either 6 samples, either 10 samples, or hundreds of samples with the same method. So we should not be limited by the number of samples that we can measure. And also, we want something that's customizable. So if we see that we need to include more metabolites in the method, we could do it. Or if we need to remove some metabolites from the method, we could do it. And also, it was quite short method time that was allocated for us. So we really needed to have something we can set it up quite fast to meet all the requirements that were set for us. So then we looked around if there's something already available that we can use as a starting point. And what we found was the Agilent Metabolomics dynamic MRM database and method. So this method and database is based on Agilent 6470 triple quadruple system coupled with LC system Infinity 2. So what this method uses for separation is ion pairing separation mode. So ion pairing is known to be a really powerful tool to separate analytes with different properties from polar to non-polar. And in this method, tributylamine is used as ion pairing reagent on the C18 column. And also ion pairing is known in the field to be quite unstable. So, and in this method, there was a lot of thought and effort put in to really make this separation method stable. So there was a specific switching valve or specific setup that allows backflush of the column. So there's a switching valve installed in a column compartment, and once the gradient is over, it switches and allows to backflush the column after the gradient. And this really improves the stability of the separation. And also, you can see here that the effective gradient is 24 minutes, so this is when the separation is happening. Then after those 24 minutes, the valve switches, and its column is backflushed with acetonitrile. And afterwards, it's again reconditioned for the next sample. And so the whole method takes around 40 minutes, or 40 minutes for analysis. And for backflushing acetonitrile is used for this method. For detection, dMRM or dynamic MRM mode is used, which means metabolites are detected and measured around the retention time when they elute from the column. And as tributylamine is used as ion pairing reagent, only negative ionization mode is possible. So this a bit limits the number of metabolites or class of metabolites you can measure with this method. But also I want to point out here that method is fully customizable. So if needed, you could include new metabolites or you could remove the methods. So, and method as comes, as it's installed, it provides analysis of 290 metabolites, and it covers a really big portion of central carbon metabolism. You have such an important metabolite like sugars, amino acids, nucleobases there. It really covers good the landscape of metabolic pathways. At this point, what I also would like to show or point out is what we unfortunately figured out a bit too late. So the system is used to analyze phosphorus-containing compounds, and everything is stainless steel. And there is a specific column passivation procedure. So basically what it means, you flush your HPLC system in your column with a diluted phosphoric acid overnight. And what it does, it increases sensitivity and method performance for phosphorus-containing compounds. So you passivate all your system. And we learned it hard way that's actually needed to do it. And here you can see the results before we did it. We really puzzled why we cannot see ATP at these high concentrations. And afterwards we saw, okay, you need to do this column passivation. And then we could see this really dramatic increase in sensitivity for this particular compound. So to sum up, this is the method, how it comes. And this for us was really a starting point, right? We said, okay, we have the method, we have the setup that allows us to detect 290 metabolites. We have a good, robust LC method. There's a basic data processing method coming with that, with indicated retention times, with integration parameters, and so on and so forth. So method as is, you could take it and really use for relative quantification. But what we wanted to do in our lab really is to bring it to the next level, to really achieve absolute quantitative workflow for this method. So this for us was a starting point actually for our work. So the first thing that we did, we really carefully evaluated the provided MRMs. So there's MRMs to detect 290 metabolites, and there's a quantifier and qualifier. And we knew that for our purposes, we don't need qualifier, so we just need quantifier. So we really looked which MRM we kick out to just have one MRM per compound. Additionally, we removed metabolites with a bad analytical performance. So those were really ones either with a low sensitivity or really with bad peak shapes. And also we created sums for metabolites that are not separated by the HPLC system. So these were sugars and sugar-phosphates mainly. Additionally, we were able to include several new metabolites. These were 7 alkyl carnitines, also folic acid and folic acid metabolites. So at the end, we ended up with 190 metabolites in the detection method. And if you want to achieve absolute quantification, you need to have calibrations. And if you want to have calibrations, you need metabolite standards. So we really looked out there and to see how much metabolite standards we can buy commercially. And we ended up with having our own metabolite standard library of 190 metabolites. So 130 metabolites we get from company Metasci in Canada. Then around 60 metabolites we got from Sigma. And other metabolites we got, either we had them in-house or we got from other sources. So we were able to really purchase every single metabolite. We had the method separately as a separate compound to really establish our own standard library in-house. So, but there's a aim for us was really to put all those metabolites into one mix, to have all of them in one vial that then could be used to set up calibration curves. But there are several, let's say, practical challenges with that. So you cannot really put them all together at once. So the one challenge is there that really, if you would just dilute them in equal molar volumes, the highest concentration you would get would be around 250, 200 micromolar. And for some metabolites, it's completely fine, but for some, it's way too low. So some metabolites are quite much higher concentration cell extracts. Other thing is solubility. So how can we really mix these analytes that have different solubilities together? So we thought about it, what to do, and the solution that we came up with was an experiment that we called 22-point calibration. So the basic idea was here that we will start for every single metabolite at relatively high concentration of 500 micromolar, and then have 1 to 2 dilution for that one. And this we would repeat 21 times. So then what we had was a concentration that would range from 500 micromolar to 0.2 picomolar, right? And this experiment would allow us to first of all assess limit of detection and limit of quantification for method, because we would really see where we lose the sensitivity or where we really approach the limit of detection. It also would give us a really very valuable information about calibration ranges, meaning we would know where our samples stand in this range of concentrations. We'd also get information about linear range, so meaning when the instrument response is still linear, and also about dynamic range. And most importantly, we'd also learn a lot about solubility. There were certain information provided about some metabolites where they are soluble, but for some it was really trial and error. So it really get a lot of valuable information just from this one single experiment. But again, the practical limit remains, right? You cannot just mix them all together, as I mentioned before. So what we did, we took all those metabolites and divided them in 4 groups. And we divided them based on their retention time. So our thinking was, if we mix them together, and then the ones that come together on a mass spec, they would influence each other similarly. So the ion suppression effects will be the same, or effects on separation efficiency will be the same, because at the end we have quite high concentration. So then we really sliced chromatogram in 4 parts, and then mixed those compounds that come in a given part together, and made this 22-point calibration experiment. So then we looked at results, and what we've seen is for more than 150 metabolites, the linear range was really good. It was across 3 orders of magnitude. And for the majority of compounds, the limit of detection was below 0.1 micromolar. So it was really good sensitive method that we had here. And here's one example that I want to show for glutamine, the results, amino acid. And this is what you typically would expect, right? That you go up with concentration, you see that there's kind of saturation effect, ionization, so the curve is not anymore linear, it kind of flattens out. However, it changes if we start to implement internal standard. So if you start to build a ratio of internal standard, it again becomes linear. So this really highlights the importance of internal standard for quantitative workflow. it not only improves the precision and accuracy, but also improves the linearity for calibrations. So then we had this calibration curve. We also ran several samples, cell samples, tissue samples, plasma samples, to really see where they sit in this calibration range, in this calibration curve. And based on this, all this information we acquired, we really set up our calibration curve, and it consisted of 10 points. The 7 points what was so-called core calibration. So this would be what would cover the whole sample that we had, or this would cover the range where the sample concentration sits. And then we had 3 points what we called service points. So these would be points to be just in case if in some sample we overshoot the calibration range. So the highest calibration, the highest service point was 2 times higher than core calibration. And also we had 2 extra levels in the lower end, just to be sure that also when there's lower concentrations, we still will be within the calibration range. So, and this is how it looks like then, without log scale. And we really try to be quite equidistant with our calibration curve to really have that it's really nicely sits in the middle of our samples there. And all the compounds grouped in 6 groups based on calibration concentration. So majority of them was from 10 to 0.01 micromolar. And this really allowed us to set up really nice and easy workflow how to mix standards together. So now we can really adapt range for every single compound separately. We can have some premixes that we take care of solubility issues, and then at the end mix everything together, and we have this one big mix that we use for calibration. And if we look at the calibration performance, so for majority of compounds, we had really good calibration performance. So good in this regard means the R squared is higher than 0.95, and we have at least 6 points in the calibration curve. Then there was around 60 compounds with really acceptable, and acceptable in this means the R squared is higher than 0.92, and we have at least 5 points in calibration. And there were some compounds where we really had some problems with calibration. And this could, or in some cases was sensitivity issues, so we were a bit on the lower range, so we had 3 points or 4 points in calibration curves, and some analytes we really start to observe some issues with the stability. The next thing was, as I mentioned before, internal standards are important, and there are several reasons for that. It's to get better precision, to get better accuracy, and also to improve linearity. And first, when we start to think about our internal standard strategy, we thought, okay, we're just going to follow the same workflow as we did before. So we screened for what are the internal standards available there for these metabolites. And we could find 43 compounds separately commercially available, but the price tag was a bit too high for us. So I said, okay, what are other alternatives? And as an alternative, we thought about using C13 labeled metabolite extract. So there's a company, it's a spin-off from University of Vienna, it's called ISTD Isoleucine, and they really sell C13 labeled metabolite extract from a C13 labeled yeast. So you have all the metabolites are in the yeast labeled with stable C13 label. We also had already good experience with some of the commercial mixes from Cambridge Isoleucine laboratories, for instance with Metabolomics amino acid mix and labeled carnitine mix. So this was the things that we thought, okay, if we take these 3 things, then we would have a really nice coverage of all the metabolites and that we have in the method. However, there again was one practical issue, which was we really didn't know what are the metabolites in the yeast extract. And second problem was we don't know how to measure them, because in instrument there is no settings for that. So as the method comes, there is only for unlabeled metabolites, right? There is no settings for C13 labeled metabolites. So what we needed to do is really to manually calculate all the MRMs for these internal standards. So what we did first, we looked in this example for one amino acid. So we looked what are the MRM that is provided in the method. Then we really looked what is the fragmentation pattern. So what is the structure formula of the intake molecule and on the fragment. Then we calculated what is the number of carbons there in the fragment and in the precursor molecule, and then added the number of carbons to the MRM to have this MRM pair for all the internal standards. And this we did for all 190 metabolites we had in the method, and we screened the yeast extract and really then selected the ones with really good performance. So what we ended up was that with 68 internal standards in total. So 44 from this isotopically labeled yeast extract, and then we had 24 from Cambridge Isoleucine laboratories for commercial mixes. And next thing we wanted to optimize was really the concentration of internal standard. So we wanted to envision that if you add internal standard sample, so you add extra metabolites in your sample. So what can happen is that you either ruin your chromatographic separation by overloading the column, or also it could affect detection. So what we did, we looked at different dilutions of internal standard. So we started with higher concentrated, it was 1 to 7.5 diluted and 1 to 30 diluted. And first thing we looked was really at the separation efficiency. What we can see here is the example of isolating leucine. So there should be two peaks, should be two separate amino acids, and the higher we go with the concentration of internal standard, the worse the separation gets. So it really kind of gets this one peak. Another example is guanosine. So here you should really have one nice peak, that we see it when we go with a lower concentration internal standard, and then peak gets split at the moment that we increase internal standard concentration. So this really showed us that the highest concentration that we can go is 1 to 10 diluted internal standards. And not only that, but we also looked what is the precision. So what is RSD of repeated injections. So in general, what you would like to have for internal standard is as a stable and performance and as low deviation between injections as possible. And here I plotted the RSDs of different dilutions. And as you can see that the lowest RSD was for the highest concentration, which is quite logical, I would say. And so we decided, okay, we're going to stick to this 1 to 10 dilution of internal standard, even though we know that we sacrifice chromatographic performance for a handful of compounds. But this is a compromise we were willing to take. So then, with this method, we analyzed several sample types, as I mentioned, and what we've seen that the method as such is positively biased towards intercellular metabolites. So we can see that we can detect more metabolites in cell extract. We can also detect around 100 metabolites in tissue extract, and in plasma we see around 70 metabolites. So in our case, it's being positively biased towards cell extract it's fine, because cell extracts will be the major sample type that are going to work in our lab. Also, we were eager to see how it compares with other methods, right? And one of the methods that also we have experienced and we have used in our core facility is absolute IDQ P180 kit from Biography. And here we really compared what are the analyses you can detect with those methods. And as you can see, there is some part that overlaps, but there's a big part that don't overlap with these methods. So this really shows that methods are truly complementary to each other. So in P180 kit, you have this big fraction of lipids that are measured that is not covered with Agilent dMRM method. On the other hand, on Agilent dMRM method, we have things like nucleotides, sugar-phosphates that are not covered with the kit. There is a fraction of metabolites which are mainly amino acids that overlaps in both methods. And if we look at the precision, so what is RSD of repeated injections, then it's really similar between two methods. And this is also not a big surprise, as both methods use this triple quadruple as a technique for detection. Also, the thing that we were concerned was the retention time stability. So here I plotted retention time of single sample across 4 batches. So every single batch consists of around 80 cell samples. Then we have calibrations, we have quality control samples, we have blanks. So it's around 100, 120 injections per batch. And here in these 4 batches, you can really see that retention time is really, really stable. So there's a serine, which comes in the beginning of the chromatogram. There's NAD in the middle and ATP at the end. And you can see that across the run, in between the runs, the retention times are really, really stable. And actually what we learned in our lab is that we can rely on retention time so much that if we see that there are some issues or shifts that indicate that something is wrong with our HPLC system. So either the cleaning is needed, or we need to replace the pre-column, or some other issues. Also, we looked at accuracy, and in the field of metabolomics, the gold standard is to use standard reference human plasma from NIST, 1915. So in this human plasma, there's a reference values for several or a lot of analytes, and in this case, we plotted the ones that were overlapping in our method. So in the gray bars, you can see what the certified value, so the value that we should detect or should measure in these human plasma samples, and the blue one that actually the measured value, the one that we got. And as you can see, for those analytes But our measures with our method, we are really, really good agreement. Also, we looked at precision, meaning how the biological replicates compare with technical replicates. So what, of course, you want to see is the technical repeatability is better than biological repeatability. So, and here I plotted the RSDs of 4 cell lines, where we had 3 biological replicas per cell line, and then I have RSD plotted in blue for 3 repeated injections. And as you can see, for most of or all detected analytes, metabolites, the RSD of repeated injections is much lower than RSD of biological replicates. And for majority of metabolites, it was really lower than 10%. This shows that precision is really good for this method. Also, as this is large-scale studies that we want to use this method for, it's also important to have this intraday repeatability. So meaning, if I have my sample measured today, and I measure them, let's say, in 1 month, do I get the same results? And here we have done this exact experiment. So we measured samples in December, then we measured in January, and we plotted the results, and also the obtained values in a really, really good agreement. So also the intraday repeatability is quite good for this method. So to sum up what we have done with this Agilent dMRM method, so we took it as is, we implemented it in our lab, and then we were able to clean out, let's say, the provided MRM database to the 198 metabolites. We were able to include 68 internal standards in the method. For data processing, we also spent quite a lot of time and effort to really optimize integration parameters for every single compound. We also optimized internal standard setup so that we selected internal standard that provides the most optimal performance for the given analyte. And we also have worked a lot to really implement appropriate calibration setup for every single metabolite. And with all this work, what actually resulted is that now, instead of relative quantification, we can really get absolute quantification for 191 compounds. And the final workflow, what it provides us is that we can analyze 96 samples within 4 days. So this is not the most high throughput assay, but as I said, the throughput is not the key here. It's really the robustness and the long-term performance. So with that, I would like to thank my colleagues at the Proteomics and Metabolomics facility, Daniela and Bettina, who did the most work on this project. Also my other colleagues from Sam, Tabea, and Vitaly, and also people from Agilent. And thank you for your attention, and I'm happy to take any questions.
And thank you, Dr. Klavins, for that informative presentation. We will now start the Q&A portion of the webinar. And if you have any questions you'd like to ask, please do so now. Just click on the Ask the Question box located on the far left of your screen. So let's get started. Let's take a look at our audience member questions coming in now. Dr. Klavins, which internal standard is used for metabolites where there is no isotypically labeled compound in the method?
Yes, thank you for this question. So from our experience, what we have seen is that, of course, in the method we don't have all internal standards, so we need to assign different compounds for different compounds as internal standards for those metabolites. And from our experience, what we have seen is that retention time plays a major role there. So in terms of accuracy and then precision. So the closer is the retention time for the compound and internal standard, the better the performance of the compounds. So in this regard, we really took the closest eluting compound as internal standard. For those ones, we didn't have isotypically labeled internal standards.
Thank you for that. And we actually have one more question from our audience. Can methods be used to in positive ionization mode?
No, unfortunately, this is one of the, let's say, main limitations of the method. So as separation relies on ion pairing reagent, which is tributylamine in this case, it's not possible to detect compounds in positive detection mode.
And thank you so much. And Dr. Klavins, I want to thank you for your presentation today. Did you want to provide any closing remarks to our audience before we close?
No, I just want to thank all for your time and attention, and also for the great questions. Thank you.
Thank you again for your time today and for your important research. And before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via contact information you provided at the time of the registration. This webcast can be viewed on demand through May of 2021. Until next time, have a great day and stay safe. Bye-bye.